And he said that tributyltin could sex reverse genetically female Japanese flounder. I said, what? So if you take this population of fish that will be 100% females, and you treated them with tributyltin when they were larvae, 30% of them would become males. So as a developmental biologist, I'm pretty interested in that. So I called back to the lab and I said, guys, can you extend the testing of TBT from just SXR and test all the other receptors? And we had, because I was at the Salk Institute, we had all those receptors in our inventory, if you will, because I was studying those when I was there. And we found out that it activated two receptors that we're interested in. One was called RXR, which is a partner for about 7 other receptors. It's a kind of retinoic acid receptor, but it functions as a partner with lots of other receptors. And the other one is a receptor that has the terrible name peroxisome proliferator activated receptor gamma. That's an artifact of history, that name. These are fatty acid receptors. But because the first chemical that was shown to activate the receptor was a peroxisome proliferator, that's where it got its name. And the name's never been able to be gotten rid of.
That's the way it usually goes.
Yeah. So it activated those receptors. So he said, yeah, okay, that's interesting, but now what? Because that's not what we were working on. That was a side project for us. It was only funded by the Japanese government. And we said, well, okay, well, let's test what it does to cells in culture. Can it make these pre-fat cells into fat cells? Yeah, it did. Okay, now what? Do we continue with this or do we drop it? Right? Because that's not our field. We didn't work on obesity and nobody wanted to hear that. I remember the very first time I wrote a grant about this, one of the reviewers wrote in the review, how dare you waste our time with such a ridiculous idea that chemicals can cause obesity? Everyone knows that diet causes obesity.